Hello, everyone, and welcome to today's live broadcast, Optimizing G Protein-Coupled Receptor Expression in Mammalian Cells. It is presented by Dean Staus, PhD, and Laura Wingler, PhD, who are both postdoctoral researchers at Duke University. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about our sponsor, visit www.thermofisher.com. Before we begin, I would like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please use the Ask a Question box to let us know that you're having a problem. Please join me in welcoming Dr. Staus and Dr. Wingler. I will now turn the presentation over to them.
Okay, great. Thank you. So Laura and I today will kind of divide and conquer and tell you a little bit about the work that we've done in the lab that's trying to express GPCRs or G-coupled protein receptors in mammalian cells for large-scale biophysical and structural work. So a little background before we get into the nitty-gritty. So G protein coupled receptors, also referred to as 7 transmembrane receptors, are characterized by 7 spanning helical regions, and this is the largest family of membrane proteins. There's approximately 800. Majority of those, however, are referred to as orphan receptors. We don't necessarily know what ligands bind to those particular receptors. So G-coupled protein receptors are very important in converting extracellular cues or signals into specific cellular responses, and so therefore they regulate a vast variety of physiological processes. And so because of this, they are actually the target of a significant number of drugs on the market. In fact, about 33% of them on the market target these particular proteins. Some examples of that, some that you might be familiar with, are things known as beta blockers. These block the receptors that bind adrenaline. Serotonin receptors are often targeted in several neurological disorders. And maybe most also very familiar are the opioids. So these are receptors that are targeted in pain. So a little bit more information on the signaling that is downstream of these particular proteins. And again, the slide says it's complex but very important biology, and this is very much true. So these proteins are fairly dynamic in nature. So they reside in lots of different states, and so in the inactive state, which was not bound to its cognate ligand, which binds to the extracellular portion of the receptor, is an inactive conformation. When bound to a ligand, such as for an adrenergic receptor would be adrenaline, for example, upon binding to the ligand, the receptor becomes activated and binds to a class of proteins called G proteins, hence why they're called G protein coupled receptors. Once G proteins are activated, they do a variety of things that activate signaling downstream of the receptor and ultimately regulate a variety of different physiologically important processes. Like any important signaling cascade, it has to be turned off, and so subsequent to G protein activation, the receptor is phosphorylated, and the second protein called beta-arrestin, shown in orange, binds to the receptor and turns off the receptor by both sterically blocking the binding of G protein as well as inducing a receptor internalization. And our lab and many others have now shown that beta-arrestin can signal in its own right. And so as you can see, the main point here is that G proteins can regulate a lot of different processes, again, making them an important drug target. So unlike most soluble proteins, the structural work on GPCRs has lagged far behind those for a variety of different reasons. One of those reasons is that they're very difficult to express and very difficult to purify. But in 2007, Brian Kobilka's group at Stanford crystallized the first GPCR, referred to as the beta-2 adrenergic receptor, and this is one that was bound in an inactive state. He subsequently crystallized the same receptor bound to its cognate G protein, and ultimately this work in combination with Robert Lefkowitz's work led to the Nobel Prize. So again, so the structural work is like far behind, and it's important to note that the structural biology is an important aspect because these again are very important proteins in regulating a variety of different processes, and so having a structural or atomic view of how the receptor function could help us to design better drugs for the receptors, and also just learn how they work in more depth. So following that critical work by Brian Kobilka's group, there has been lots of now different dozens of GPCRs that have been crystallized using similar approaches that the beta-2 adrenergic receptor. But that said, remember this is a very, very large family, and so still the number of GPCRs have been crystallized relative to how many numbers there are in the genome is still relatively small. So how do people typically express GPCRs? Well, traditionally people have used insect cells. These are called SF9 cells. So there's a variety of different reasons why you want to use them. So the expression, and this is the system that nearly all GPCRs have been expressed into to date, at least that have been used for large-scale structural and biophysical work. The benefit of the system is that it's very scalable, and so you can grow in fairly large volumes. And again, these GPCRs don't express at tens of milligrams per liter. They usually express hundreds of micrograms to low milligram quantities in a good situation. And so these cells allow you to scale proportionally, and because they grow in suspension. They don't need any specific temperature or CO2 requirements. They just grow at normal room temperature. So the cost of shakers is minimal compared to other traditional systems. The infection methods are usually baculovirus based. There's very little lipid-based transfection protocols are available. So majority of people use baculovirus-mediated infections of these particular cells. So why not use insect cells? Well, for anyone that's used insect cells, the variability can be extreme. There's every time the viruses that you use to express these particular proteins can vary, as well as just the overall variability is quite substantial. I think the biggest thing for our field is, and for Laura and I, is the idea that the protein functionality of GPCRs when expressed into these cells is not great. And then one example that we have listed here is again the beta-2 adrenergic receptor. And so this particular receptor we express usually in SF9 cells, and you purify. If you look at the expression of that particular receptor, it yields about 25 milligrams, for example, from the say from 6 liters. And when you purify that and you actually measure the functionality of that receptor after purification, it's much lower, and we measure that by looking at the ability of its ligand to bind to the receptor. And so the functionality decreases substantially, and so you have to use these secondary affinity chromatography steps to get the pure and functional material. And all this is quite labor intensive, and again, you have to develop different techniques for each individual receptor. So again, in terms of functionality, there's just the functional versus non-functional. How do you separate those two after purification? This is an example of one of the receptors that we've worked on, where if you just look at the ability of the ligand, a ligand to bind to the receptor, can vary substantially between mammalian cells and SF9 cells. And so this is a simple competition radio binding assay, where we look at the competition of a radio-labeled antagonist for the receptor, and basically, as you see on the x-axis, the ability of a cold competitor, an agonist, to compete off that hot ligand. As you can see, when the receptor is expressed in mammalian cells, the affinity of the ligand is quite good, and when you express the same receptor in SF9 cells, this would be the insect cells, you see about 100-fold reduction in its ligand affinity, suggesting that just potentially the membrane environment can have substantial influences on the receptor function. So another thing what Laura will talk about more in a little bit, and I think that's something that's very, very important nowadays, is that in mammalian cells you have what we would call native post-translational modification. So glycosylation can be very important for protein function. The lipidation, efficient GPCRs, is very important. Phosphorylation, all these other different post-translational modifications can be very important to receptor function. And so again, these are kind of lost in the insect cell culture system. So I think with that, I will turn it over to Laura, who will tell you a little bit more about the exact system that we've now almost exclusively switched over to for expressing and purifying GPCRs.
Thanks, Dean. So the main alternative to insect cell expression for GPCRs has been pursued is mammalian cell expression. So there's a lot of obvious reasons that mammalian cells would be preferred to those from lower organisms. The most obvious of which is simply that these cells are designed to express GPCRs, so they have all of the proper processing machinery. In contrast, invertebrates only have a single GPCR gene, and mammalian cells, as Dean already told you, have hundreds. So in many cases, the difference in expression systems has a dramatic impact on the overall functionality of the protein. So going back to the example of the beta-2 adrenergic receptor that Dean already mentioned, as he said, when we purify this receptor from insect cells, only about a quarter of the protein is properly folded and able to bind ligands. In contrast, when we express the same receptor, one of the most common model systems of GPCRs, when we express it in mammalian cells and purify that, the specific activity of the protein is close to theoretical. Basically, all of it's properly folded and able to bind ligand. There are also a lot of other advantages to mammalian expression. So, as Dean was just saying, the fact that the post-translational modifications are similar to that would be found on endogenously expressed proteins. And then there's also other technical aspects that are convenient, such as the fact that that there are a lot of available methods for transient transfection of cells, and we'll discuss that a little bit more in a little bit. So why have people shied away from the use of mammalian cells for GPCR expression? Well, there are a number of technical problems that have stood in the way. So we'd like to take you through a few of these problems and what we've found to be potential solutions to them. So the first problem is simply that high-level expression of GPCRs tends to be rather toxic. And this probably is not a huge surprise because GPCRs are signaling proteins, and so when you overexpress them, you're going to be constantly activating all kinds of pathways in the cells nonstop. As a result, if the cell can survive this stress at all, it's going to be doing everything it can to downregulate the receptor expression. And in addition, of course, you can have activities at the level of protein folding and processing. So what's the solution to this? One answer is to use an inducible expression system so that you can turn the expression up to high levels for a relatively short time, immediately before the cell harvest. This was first applied to GPCRs quite a few years ago now by the Corona group, and they demonstrated that mammalian cells could be stably transfected with both the tetracycline repressor and with constitutively active mutants of rhodopsin that were under the control of a TET operator. This stable cell line could achieve high levels of rhodopsin expression when induced with tetracycline, as this graph from their paper shows. In contrast, when they tried to isolate stable lines for these mutants under the control of a constitutive promoter, they were unsuccessful repeatedly. Another problem with mammalian expression has been cost efficiency. Generally, the media is more expensive on a per volume basis, and the cell density of the cultures, even at saturation, is substantially lower than can be achieved with insect cells. However, we found that some of the recently developed mammalian lines that are selected specifically for the ability to grow to very high densities, we found that the increase in expression as a result of this has been able to can offset the increased cost per volume. And so on this slide, we're showing data that we obtained with the XV293F system from Thermo. We transfected 8 different GPCRs along with the tetracycline repressor that I just discussed. And when we induce the expression with doxycycline, all of them are expressing in the milligram per liter range. And as Dean was saying before, for a GPCR, this is quite high and quite sufficient to be able to pursue biochemical or structural work. Let me also take this opportunity to point out some of the advantages of using a transient transfection expression platform rather than the baculovirus systems that are predominant with insect cell expression. This is particularly advantageous for projects involving mutational analysis of a protein or where you might need to screen many different constructs, as is often common in crystallography projects. What we have found to be really lovely about transient transfection is that we can transfect a number of different constructs into suspension cells in parallel on a very small scale, for example, in just a 6-well plate. And this scale is quite sufficient for us to be able to validate the expression levels of our receptors and to perform some very basic functional tests, like some of the ligand binding assays that Dean was showing you. Then, once we validated our constructs, we can use the exact same suspension cells in the exact same transfection protocol and simply scale up to on the order of a liter to actually be able to purify the receptor. As shown in this timeline, what's really wonderful about this is that we can go from having the DNA for a new construct to having validation of it in just a few days. And then we can go to a large-scale run in just a few days more, and that's shown in the right-hand column. In contrast, the turnaround time is substantially longer for baculovirus systems that are used for insect cells. And this is because you typically have to do multiple rounds of amplification to get the virus up to high enough titers where you can get good expression. So instead of a few days, it takes a few weeks to get initial data on a new construct, and there's a much greater upfront investment of both time and resources. This difference in the turnaround really increases your ability to screen constructs in high throughput. So here I'm showing data that we generated where we had to screen about 2 different dozen receptor constructs for a project. And if you have any experience at all with baculovirus production, you can imagine how painful it would be and expensive to test this many viruses. So instead, with the transient XV293F system, which we used for this, it was feasible to test all these constructs in one shot in just a few days and then narrow it down to the few best ones for follow-up work. A final issue that has prevented people from adopting mammalian expression systems for GPCR biochemistry and structural biology has been some of the PTMs, the post-translational modifications that come along with them. In general, we consider this to be a plus because these modifications are very important for protein function, and we prefer for them to be as native-like as possible. But there are some downsides. and one in particular has been glycosylation. So in mammalian cells, glycosylation tends to be more complex and more difficult to remove enzymatically than it is in insect cells. The glycosylation pathway consists of a number of steps. It begins with a common core that's added, and then it's elaborated by numerous enzymes. But due to the number of steps in the formation of end-linked glycans, which is the class is predominantly found on GPCRs, the end result is a very heterogeneous mixture of glycans. This can be an issue for applications such as crystallography, where you want to minimize the heterogeneity of your protein samples. So, several approaches have been developed to stop the addition of glycans earlier in the pathway, before the species, while it's still at the step of having this glycan core, before the species begin to diverge. So the first approach is by genetic inhibition, by knocking out a glycosyltransferase. This again was first described for rhodopsin, also by the Khurana group, several years back. And what they did was that they created a stable HEK line with a GNTI phenotype, that is the N-acetylglucosaminotransferase 1, which, and by knocking this out, they blocked the processing of end-linked glycans at the second step. And as you can see in this figure from their paper, when they expressed rhodopsin in the system, its migration on the gel changes from being a very broad heterogeneous band, shown in lane 2, to a very defined species, which is in lane 3. The same trick of using this GNTI knockout HEK line has also worked in our hands for other GPCRs that are less tractable and more refractory to expression than rhodopsin is. So the gels on this slide compare the purification from a typical native-like HEK suspension line to that of a GNTI minus line. And you can see that the purified protein in the far right column of each gel, that for the native-like one on the left, it's very faint because it's a very spread out band, whereas on the right one from the GNTI minus line, it's a much more defined species. But what if you're restricted to the use of particular cell lines that don't have the GNTI minus phenotype? Well, there's another option to restrict glycosylation early in the processing pathway, which is pharmacological inhibition of the glycosylation pathway. One example of this is kifanencine, which is an inhibitor of alpha-manosidase 1. So this is actually the step before the GNTI that's been knocked out on these cell lines that I've talked all about. Again, this is stopping the glycan processing before it's elaborated past this defined core. So this slide shows the results when we purify GPCRs from XV293F cells that are either untreated or that are treated with kifanencine. So once again, if you look at the gels, in the untreated cells, most of the receptor is actually part of a large smear, which you can see in the final step from the SEC. In contrast, when we treat with kifanencine, we end up with the receptor migrating as this very defined double band. You can also see the difference on the size exclusion chromatographs of these two receptors, which I apologize. The labels are reversed on that. The red line actually represents the untreated, which you can see is a much broader peak than the black band, which is the kifanencine treatment. So where the peak becomes much narrower and tighter due to the homogeneity of the prep. Another nice thing about using these restricted glycosylation, either from GNTI minus lines or from the pharmacologically treated ones, is that the glycans become much more sensitive to deglycosidases. Generally, to fully remove the glycans from receptors enzymatically with enzymes such as PNGase or EndoH, they have to be denatured. In contrast, oftentimes with this restricted glycosylation, the native proteins can be deglycosylated very efficiently, and they're still in their native form by these enzymes. So this Western blot shows that, and in the first lane, what you see is basically the receptor that was not treated with the cifenensine or with the EndoH enzyme, and so it's very broad until you deglycosylate with PNGase, which is shown in that bottom panel. So then, if it's denatured, it will collapse to a single band. If you treat with cifenensine, the top gel shows that the receptor is initially a much more defined band. But even when we deglycosylate with EndoH, the non-cifenensine treated sample does not collapse into the lower band, whereas the cifenensine treated material will. So another thing to note, this is kind of a word to the wise, is that different deglycosylation methods can actually have different impacts on the functionality of G protein coupled receptors. And so I mentioned that there are a couple of different enzymes that can be used to finish removing glycans from GPCRs with restricted glycosylation. So PNGase-F actually removes all glycan species, cutting the bond between the asparagine and the first residue. And this actually converts the asparagine to an acidic aspartate residue. EndoH, on the other hand, leaves the first glycnac residue and removes the rest. So what we found with one of our receptors, quite surprisingly, is that if we grow this receptor in the presence of cifenensine to restrict the glycosylation, that gives us a much more homogeneous band, as shown in the gel up top. And in fact, it has no impact on the ability of our receptor to bind ligand, as shown in the bottom graph. And so you can see that by the fact that the black lines and the green lines are completely on top of each other. Likewise, when we use EndoH to remove the remaining glycans, leaving only that last glycnac residue, that also has no impact on the ability of the receptor to bind to the agonist. In contrast, when we use PNGase-F to completely remove all glycans from the protein, that lowers the affinity of the agonist for the receptor by about an order of magnitude. So the point being that it's really, all these modifications are really important to validate with your specific receptor in order to obtain the most highly functional protein at the end. So in conclusion, I hope that we've convinced you that in many cases, mammalian cells are a very viable alternative for the large-scale expression of GPCRs. There are ways to overcome most of the issues that have really plagued the field to date, including circumventing the problem of toxicity by using inducible expression systems. We can use a lot of the transient and transfection systems that are now available enable us to really start doing much more high-throughput screening of constructs than was previously feasible. And finally, some of the issues such as glycosylation and the issues it can bring up for structural biology can be avoided using either genetic manipulation or pharmacological inhibition. So that's the conclusion of what we had to present, and we're happy to open it up for questions now.
Well, thank you, Dr. Staus and Dr. Wingler, for your informative presentations. Yes, we will now start the live Q&A portion of the webinar. So if you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, are there strategies to help expression using chaperones, etc.?
Sure. So basically there are strategies to stabilize the receptor from both ends, we could say. So one thing that often helps improves expression is to express in the presence of an antagonist. So a ligand that will basically, this again is kind of gets back to the toxicity issue. Lots of times even the basal expression of a receptor can be causing problems for the cell. So in many cases, putting an antagonist into the culture media to basically shut down that signaling can really increase the expression levels. So that's stabilizing the protein sort of from the outside, you could say. Another thing that has been done is co-transfecting receptors with some of their transducer proteins, like heterotrimeric G proteins. So in some cases, people have actually been able to purify entire complexes of GPCRs bound to a transducer. This was done for a couple of cryo-EM structures of receptors with G proteins in recent days. So yes, both of those have been explored as possibilities.
Great, thank you. Are there any mutagenesis strategies for improving expression of GPCRs?
So that has actually been explored by a number of groups, and there's actually entire companies founded on that principles. So usually the strategy is just to do random eugenesis throughout the receptor, and then do high throughput screening to identify mutations that improve expression. So we always the problem with mutagenesis is that it always kind of leaves a question mark in your mind. You know, why exactly did this mutation increase the receptor expression? And lots of times it's because it's actually having an adverse impact on the ability of your protein to signal. So basically, it's less toxic because it was there was less constitutive activity. And so when you start doing structural work, biochemical characterization, pharmacological characterization, you know, there's always some concerns associated with that you know, that it's not really as native-like as it could be.
Well, great, thank you. Do other tags besides FLAG give a reasonable level of purification from these cells?
So we almost exclusively use FLAG tag for purification. We have used like a 6X his tag, but in our experiences, those things that are like a nickel, a his tag, it just tends to be a lot more non-pure at the end of purification. So we tend to use FLAG tags just because the purity is that much better. So we haven't explored a whole lot of tags, but I would say a head-to-head comparison between FLAG and his, FLAG is far superior.
Well, thank you. Okay, your next one is, in your opinion, why do you think HEX cells, the gold standard for cells, were not able to express melanocortin receptors or its ligands, but the C20A4 cell line chondrocytes were able to express these receptors, melanocortin receptors or GPCRs? What do you think could have gone wrong?
Well, so let me start off by saying that while we've had a lot more consistent success with HEK cells than with, say, insect cells, with the GPCRs that we've tried so far, I certainly can't make any guarantees that it's going to be a universal, a 100% universal solution. So regarding these specific cells, I don't have a lot of experience with the other line that was mentioned, but it could be something as simple as different. Each cell line is going to have somewhat different post-translational modifications, a different complement of proteins such as that, and so there could be even within mammalian cell lines in general, there are probably going to be a lot of very cell type specific differences.
Thank you. Okay, next one is, how about BACMAM systems? Is it a nice balance between these two systems to overcome DNA gigaprep costs? So in many cases, a BACMAM system is going to have a lot of advantages. What's really been nice for us in terms of the transient, as I mentioned before, is simply the fact that we can screen so many different constructs in parallel. Once you've narrowed it down to a few, a BACMAM system can have a lot of advantages since you're only dealing with one or two. I will say that that really with the working at things in the liter scale, you shouldn't be having to do gigapreps at a time. You know, lots of times a simple maxi prep will see us through all of our needs for a particular construct for a run or two. The next person says, thank you so much. After the purification with denaturing conditions, is it necessary to do other procedures for the protein?
So first of all, let me say that we do not typically use denaturing conditions when we purify. Usually we want to make sure that the proteins stay natively folded. We didn't really get into it today, but purification of GPCRs usually is sort of an art field unto itself. Once you do get up to high expression levels, and there's a lot of work that's been done in regarding different detergents that can be used to help preserve functionality and that kind of thing. But that's kind of a different topic for a different day.
This next one I'm going to paraphrase. I'd like you to help me with it. Can you not perform lipidation on insect cells?
Right. So insect cells do. So for example, a lot of GPCRs have palmitoylation sites, and insect cells do palmitylate it. But usually, in our experience, the percentage of receptor that gets correctly lipidated is lower than it is in mammalian cells.
Okay, great. And excuse me if I mispronounce a word here, but do you recommend mammalian cells for BRET, B-R-E-T assay?
Yes, usually for BRET, you're not dealing with needing large quantities. It would be a smaller scale 96 well plate assay. And yes, usually those assays are typically performed in mammalian cells because they're looking at signaling and interactions between proteins. And so you basically want your normal native complement of receptors around.
Okay, how about screening in HEK cells and scaling up in SF9 cells?
Well, in some cases, this might be effective, but as Dean kind of showed you earlier in the presentation, a lot of times we see very different major differences in the functionality of the protein going from SF9 cells to insect cells, such as the ability of basically the affinity of the receptor to bind a ligand. So this kind of points to the fact that in many cases in SF9 cells, your receptor is simply not being properly expressed. We've also seen very construct-specific effects going from one cell type to another. So a construct that expresses great in SF9 cells doesn't express in HEK cells, and vice versa. A construct that expresses great in HEK cells doesn't do so well in insect cells. So it doesn't seem to be a very good correspondence from one cell type to the next in terms of what does best.
Okay, well, thank you. And if additional questions come in, we can jump over to them. But in the meantime, if there are no more questions, I would like to once again thank Dr. Staus and Dr. Wingler for their presentations. Do you have any final comments?
No, I don't believe so.
Okay. Well, I would also like to thank Thermo Fisher Scientific for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May of 2019. Please share with your colleagues who may have missed today's live event. And also stay tuned for our next presentation, Efficient Purification of Recombinant Strep-Tag Fusion Proteins from XB Supernatants using Streptactin XT. It is presented by Dennis Niermeyer, MS, a scientist at IBA Life Sciences. You won't want to miss it. That is all for now. Thank you. Oh, wait a minute. Hang on one second. All right, it looks like another question has come in. I just want to take okay. So one more question has come in, but it's a very complex question, and we will take that one after the presentation. It's very complex, very technical, and we'll take that one after the presentation. So thank you again. That is all for now. Thank you for joining us. We hope to see you again soon. Goodbye.